Good afternoon, everyone. I'm Marissa Ochoa, and today I have the pleasure of introducing Dr. Amy Angert. Dr. Angert received her PhD in plant science from Michigan State University. Her dissertation work focused on the ecology and evolution of elevation range limits in monkey flowers, and Dr. Angert now continues to focus her research on the evolutionary ecology of species geographic distributions at the University of British Columbia. Her lab utilizes experimental manipulations in the field across diverse habitats in Western North America and in the lab with observations of natural populations and tools from quantitative genetics and physiological ecology. Dr. Angert's research seeks to answer fundamental questions at the interface of ecology and evolution by examining what limits adaptation at the edges of species ranges, why closely related species vary by orders of magnitude in range size, and how ranges are likely to shift in response to climatic changes. Today, if you have any clarification questions during today's talk, please feel free to use the Q&A function. If not, please save your questions for the end and we can use the Q&A box, or you can raise your hand and we will unmute you. Thank you. And with that, here's Dr. Angert.
Thank you very much for the introduction and the invitation. I'm really excited to share with you guys some of the work that my lab's been doing. Let's see. Good. Are we good? Does everybody see the slides?
Yes, okay, looks good.
Thank you. Great. Okay. Well, after that really nice introduction, I probably don't need to give you guys an overview, but I did want to start by just sort of giving a picture of the kinds of questions and the work that my lab does. And the disclaimer is that it's because I'm not actually going to talk about any of this stuff today. But what we do in my lab for the most part is we focus on species geographic distributions. We stare at these blobs on the map that sort of depict the geographical distributions of species. And we think about things like, what causes that margin that defines the range limit for any given species? Is that simply a transient boundary moving as the species disperses, or is it an adaptive limit? And if it's an adaptive limit, why, given that most species have locally adapted across broad swaths of space, why would adaptation suddenly fail at a species geographic range limit? And so in this way, we can use range limits as sort of a way to study the more general problem of sort of why we see niche evolution in some cases and niche conservatism in others. And although every species has a range limit, even closely related species can vary by so many orders of magnitude in their range sizes. And so what is it about the eco-evolutionary dynamics of dispersal and adaptation that would cause some species to be narrowly endemic and others to be broadly distributed across large swaths of the landscape and even invasive in other places? And I would really, really happily spend my career studying what I admit are these sort of very esoteric questions about limits to adaptation. But the truth is that pretty much every empirical data set that I've worked with or gathered myself has borne some sort of greasy thumbprints of climate change on it. And I can't ignore the fact that the ranges that I'm studying in real time are in disequilibrium with the environment. So ranges are shifting. And increasingly, I'm sort of focusing my lab on questions about the process of dispersal to track suitable climates and rapid adaptation as strong selection is sort of moving species out of equilibrium with the environment. What are the limits on adaptation in the form of evolutionary rescue that might forestall, say, range contractions? So these are the kinds of questions that we tackle, and the systems that we do them in are focused on sort of three pillars. The first is using monkey flowers, primarily in California, so I'm sure many of you know these species. Doing field experiments and observations across species ranges to think about the eco-evolutionary dynamics of range limits. Since moving to British Columbia, I've been working in the Washington and British Columbian Cascade Mountain ranges, resurveying historical vegetation plots, experiments to dissect sort of who and how species are moving across the elevation gradients. And then most recently, we've been working with mesocosms in the lab and cosmopolitan duckweeds, where we can actually create ranges on a much more tractable scale with a linear thermal gradient and watch either a single species or assemblages of species evolve under climate change scenarios along these mini ranges and lab scaled experiments. So I say this because this is really what my lab does. We're really focused on fieldwork and experimental manipulations. And I'm not going to tell you about any of this, and I'm happy to chat with those of you I'm meeting with afterwards about this work. What I am going to talk about is a detour that we took a few years ago with a data synthesis looking at local adaptation. Now, it was prompted by a couple of field experiments that we did have ongoing at the time. And so the first one was an experiment that Chris Muir, who was a postdoc at the time at UBC, was doing. He'd done a couple big common gardens in California with the scarlet monkey flower, one in the south in San Diego County, one in the north up in Plumas County. And in each of these gardens, he'd planted genotypes representing an array of populations that came from across the species range. And the blue and red here is showing that he had a drought and a watering treatment within each garden. I won't talk about sort of the motivation or why he was doing it, but what he did was he brought some preliminary results into our weekly meeting after he'd come back from the field. And these are results where he was looking at our best estimates of fitness for each of these populations in each garden. And so it's going to be set up like this. We're on the x-axis. We have the source populations he'd collected from the field and planted into each garden, southern garden on the left, northern garden on the right. And then the y-axis is our best proxy for fitness. Now it's a short-term estimate of what is actually a long-term and difficult to measure thing, but it's our, a component of reproduction of the plants in the gardens at the time that he brought these preliminary data in. And our expectation, if populations have evolved adaptations to do really well in their home sites, then you would expect that in the southern garden, it's the south populations that should have had the highest fitness. And in the northern garden, it's the northern populations that should have the highest fitness. And I put a star for where you would expect to see the highest values in the data. And so we looked at the data for the southern garden. Isn't that beautiful? As you move north, the populations that came from further away and further to the north were doing worse and worse. And the southern populations that were local to the site had the highest fitness. And then we looked at the data for the northern garden, and it's a little bit puzzling. So now we have the same sort of rank order, but it's the northern populations are still underperforming, even though now they're at home, right? And so we sort of explained it away, it's a long-lived perennial, Chris. This is just the first year of fitness data. What it means is that you just need to keep at it. I'm sure over time they'll catch up. Perhaps there'll be a cold snap, something that will make that advantage of the local adaptation apparent and measurable. And so this was, a little bit odd, but we sort of put it out of our minds because the experiment was only in its first year. But later, in literally the same week, I had a meeting with another person in the lab, and this was Megan Bontrager, who was a PhD student at the time. And she's working on a completely different study system, this gorgeous little annual Clarkia pulchella, a totally different part of the world up in the sort of Okanagan region of BC and Washington, sort of dry sage steppe. And so she had done another common garden at these two sites, these focal sites, F1 and F2, where she had gathered all of these genotypes in purple and pink colors from across the range and planted them into this northern common garden at the northern edge of the species range. And so she had data from her field season as well. And it looked like this. And so when we look now at lifetime fitness as a function of whether you were the local population on the right, those F focal sites, or any of these other populations that had been transplanted in, you see there's no average difference, no local advantage on average. And in fact, many of the foreign populations were doing much, much better than the local. And now in Megan's case, this is a tractable little annual plant. She doesn't have to keep waiting for more fitness data. This is it. This was their lifetime fitness, and there was still no evidence of local adaptation. And so having both of these results come in the same week was really, really perplexing because, the thing you learn as an evolutionary biologist is that local adaptation is everywhere, right? We have these canonical examples of plants along elevation gradients or color matching of animals and insects. You can probably think of your favorite example of local adaptation because these are sort of what we grow up with, learning about the magic of evolution to craft genotypes to fit the environment, right? And so, local adaptation is ubiquitous, but my lab just is really bad at measuring it. What is going on? And so we turned to this data synthesis or meta-analysis that Joe Herford had done in 2009, where he compiled all the studies that people had done to try to detect and quantify local adaptation. And so in this sort of two-dimensional space, the y-axis is the fitness advantage when you're local, which you would expect to be high, and the x-axis is the fitness advantage when you're foreign, which you would expect to be low if there's trade-offs where the adaptations that make you fit at home actually have a cost in other environments. And so local adaptation is sort of this upper left quadrant, and you can see that most of the data points do reside in that quadrant, right? But these other regions of space where populations either do poorly everywhere, even at home, or they do really well everywhere, even when they're not at home, there's actually quite a bit of data in those other quadrants. And so yes, local adaptation is prevalent, it's ubiquitous, but it's not universal. And what we wanted to do was sort of try to ask, well, what does explain why some study systems or some populations end up in these other quadrants as opposed to being classically locally adapted? And so we went on this sort of foray into the world of meta-analyses, and that's what I'm going to tell you about today, because we've what was supposed to be a quick side project turned into several years of effort. And so I'm going to tell you about this database that we assembled of field transplants that that quantify local adaptation. I'm going to tell you about two projects, one looking at how local adaptation varies over time, and particularly with climate change, and another that's talking about or examining how local adaptation varies across space. And I'll sort of present the hypotheses and predictions for each of these within the modules. Okay, so to assemble the transplant database took a lot of work by a lot of people shown here. These are undergrads, grad students, and postdocs at the time. They've all since moved on, but it really was spearheaded by Megan von Traeger, who did essentially a second thesis worth of work spearheading this major effort. And so what we did was we started with a literature search and had about 2,000 studies to screen based on our search terms. Many hundreds of those we actually screened quite carefully by reading abstracts and methods. Got to a set of a little over 200 that met all of our criteria and for which we attempted data extraction. But unfortunately, because of inconsistencies or missing reporting in terms of locations of populations or sample sizes or whatever, we got down to about 150 studies for which we actually could extract all the data we needed, and that represents about 160 some species. So here's the warts on the data set. So although our search was agnostic to study system, of course it's biased towards plants, and of course it's biased towards short-lived plants. So most of the data set was sort of short-lived herbaceous plants. And again, although our search was agnostic to location, of course it was biased to the Northern Hemisphere. And you can see here in green, the populations that people sampled from, and in purple, the destination sites where they were transplanted. All in all, though, many, many hundreds of sources and sites. Okay, and in terms of fitness estimates, because to quantify local adaptation, we need an estimate of fitness. This is showing you the breakdown wherein, for the many, many studies, we only had early, say, initial survival as the best estimate of fitness. In other cases, we got reproductive components of fitness. And in about half the cases, we could get something like a lifetime composite across sequential demographic transitions that got us towards lifetime fitness. Okay, so these are the data we're working with. And let me talk now about why we were interested in variation and local adaptation over time. And to do that, I'm going to return to one of the initial studies that sort of inspired this effort, and that was this Clarkia study, where we had the foreign populations on average doing no worse than local populations, and in fact, sometimes quite a bit better. And when we sort of think about, well, who are these foreign populations? Where are they coming from? We can look at them in climatic space. And so this is a bivariate plot of precip and temperature, sort of ordinating the populations based on their climate, historically, shown in the colored circles. And so the transplant sites are these F1 and F2 sites that were sort of intermediate in temperature historically. But in the year that Megan did the transplant, it was actually anomalously hot. Right, so over here. And in fact, the conditions during the experiment were better matched to some of these southern populations than they were to the local populations' historical climates. And so when we separate out the performance of populations based on whether they were coming from someplace warmer than the transplant site or cooler, Now we have a lot of that variation explained, where the high fitness of the foreign populations is coming from these pre-adapted populations from hot sites. Okay, so it's these warmer populations that were outperforming locals, not just any foreign population. In fact, the cooler ones did numerically worse. And so our hypothesis then becomes that maybe populations were historically local adaptation, but climate change is sort of pulling that historical climate out from under them to which they were adapted. And now they're sort of bleeding out into these other corners of parameter space, where now maybe the local population isn't doing well anywhere. Foreign populations coming in are distantly adapted or maybe good across a range of sites. And so what we wanted to tackle with this meta-analysis then was to ask, well, really, can we use transplant experiments to assess how robust fitness of each population is to variation in climate, whether that's spatial or temporal? And then is local adaptation weaker or less prevalent as climate anomalies increase and as time goes on? And so I've told you that we've started with these transplant studies, and what we're doing is extracting from them our fitness data. And we're calculating then relative fitness, relativized to the mean fitness of the species at the site, as well as local adaptation, which is the relative advantage of one population over another. So let me walk you through that with this example. So this is one of the species in the data set. This is a white pine. And this is a pretty common design where it's not a single site common garden, but it's not a fully reciprocal transplant either. So we've got two transplant sites, one in the north and one in the south. And in each transplant site, they've planted in a range of source populations. And those are these colored dots on the map that are all grown together in a transplant site. And so to calculate local adaptation, we're going to rely on this local foreign contrast, which is basically the response ratio of the performance of the local population relative to any foreign population in a given transplant site. And so for this red transplant garden, we can ask if the fitness of the local population is greater than the purple or the blue or the green, and et cetera. So we get multiple local foreign contrasts depending on the number of source populations to compare to the foreign. Okay, so that's sort of the fitness and local adaptation calculations. The other thing we needed to do was for each source and site extract climate records. And so that was based on getting the lat long and going to climate products from which we could calculate the historical climates as well as the difference between historical and experienced conditions, which we're going to call anomalies or deviations. And so we rely a lot on these climate NA or other analogs that were originally created by Tongli Wang and Sally Aitken at UBC, and they've since been broadened worldwide. And they're really lovely because they downscale gridded climate data based on digital elevation models and adiabatic lapse rates, so that in even topographically complex regions, you can get a pretty good sort of point-specific estimate of historical climate. So we did that for each transplant source and site. And what we really need to do is think about how does the climate the population experienced in the transplant differ from its historical climate and historical selection regime. Okay, so here's an example to show you. So we defined a reference period, which was the 1951 to 1980. We used 1951 as our cutoff because before that, the climate records are really much too sparse. And much past 1980, on the other end, we're getting into sort of too much of a signal of contemporary warming. And so we defined this as our historical average, 51 to 1980. And then we extracted the records from the year or years over which the experiment was conducted and called the climate deviation the difference between those two. And the expectation here is that these deviations up and down from your baseline average should affect your performance. So just as a cartoon example, if you imagine this blue population transplanted at home, there is some interannual variation in temperature. Here's the historical average. And let's assume that as you move away in either direction, cooler or warmer from that historical average, that your performance might decline a bit. Okay, so depending on the year in which you do your experiment, you might have relatively high growth or somewhat lower growth, just depending on the conditions of the experiment relative to the historic average. So this is true within a site, but of course this range of variation is probably quite small. And when you harness the power of these transplant experiments, though, that are moving populations across the landscape, you get a much bigger range of climate deviations across which to assess fitness responses. So now if we have a second site, say it's a southern red site with its own temperature profile that on average is warmer, if you plant the population from the blue site in these red conditions, then we'll have a much better way to estimate sort of the tails of this performance curve as it gets into increasingly unsuitable conditions. Okay. So the relative performance, to think about things like local adaptation, we're interested in the relative performance of the local compared to the foreign population in a site. And the idea here is that really the outcome is going to depend on the conditions during the experiment. So if you imagine that the experiment was done under conditions just like the blue population's historical average, it should have a strong local advantage indicating local adaptation, right? But if you do the same exact experimental design in a really warm year, now perhaps you're going to detect foreign advantage because the conditions are favoring the performance of this foreign population. The other thing we can do here is we can estimate, even when a foreign population is at its own historic average, is there still a difference between being local and being foreign? And that difference we can assume is the local adaptation due to all the other things, right? The pathogens, the competitors, the non-climatic benefits of being local. Okay, so we modeled these response curves of fitness to climate as Gaussian curves. We assumed there's a maximum fitness that could differ between foreign and local. And we assumed that with deviations from that peak optimum temperature, we're declining on either side of this Gaussian curve. And we assume that there's sort of the breadth of that curve is the rate at which fitness declines away from the optimum. So we're sort of modeling this with the empirical data from the transplant studies. And here are the data. So here are the actual fitted curves to the real data. So we have relative fitness on the y-axis. We've got our temperature deviation on the x-axis. So that's, again, just the difference. Apologies for the difference in the lingo. This is just the deviation between the historical average and the experiment conditions the population experienced. If it was a population at home experiencing sort of year-to-year variation in weather, that's the light gray. And if it was a foreign population that's been moved across a much broader range of conditions, and those are the dark populations or the dark dots in black or dark gray. So when you're experiencing a warm deviation, you're right of zero, a cool deviation, you're left of zero. And what we can see here is that fitness does have this sort of unimodal response to temperature, where you peak when the temperature is quite close to your historical average, and you decline as you move colder or warmer from that historical average, okay? One thing to note is this really interesting separation between the local and the foreign populations in the height of their curve. So this is the sort of home site advantage that is not, in this case, attributable to temperature, to all the other things, okay? And then the other thing to note about this is that although, yes, fitness declines with strong temperature deviations, it's actually, in my mind, remarkably robust. So we're here 10 degrees different from your historical average. Relative fitness is still actually quite high. Okay, so it is a declining curve, but it's actually quite broad across very, large swings in temperature. Now, when we look at the fitness responses to precipitation, we see a completely different pattern. So these are no longer unimodal. We just see a slight trend for sort of wetter is better across the range of conditions. Again, the separation between local and foreign for the fitness advantages that are not attributable to precipitation, but no evidence that falling away from your historical local is actually detrimental. So for the rest of the results, I'm going to focus in on the temperature because the evidence for precipitation mattering is just not that strong in these data. Okay, so I want to turn away from relative fitness and thinking about the local adaptation as sort of this local foreign comparison across temperature. So Although it's intuitive to think that local adaptation will go away with anomalies, that's actually only half of the story. So it depends on the direction of the climate deviation relative to the historical climates of the population. So we talked before about how a warm deviation might favor a warm adapted southerly population. Of course, that's possible, but a cool deviation the other way could actually enhance local adaptation because it would make the warm adapted population even less adapted in this cooler site, right? And so we have to account for both the historical climates of the foreign and the local and the direction of the deviation before we can make a strong prediction or sensible prediction about what climate variation is going to do to local adaptation. It could either reinforce it or it could counteract it. And so when we look at the data separated out by whether the temperature anomaly was reinforcing towards the local population or counteracting towards the foreign population's historical temperature, we do see that they're affecting local adaptation in the ways we would predict. So local adaptation is on the y-axis, the temperature anomaly during the experiment is on the x-axis, and as the temperature anomalies get bigger, we get either more local adaptation or less, depending on whether the temperature anomalies are reinforcing. So they're enhancing local adaptation when they go in the direction of the local population's historical climate. And they go away from local adaptation. They actually cause less local adaptation when the temperature anomaly moves the experiment conditions towards that of the foreign population's historical climate. Okay, so now we can see that temperature anomalies are having strong effects on local adaptation, and those effects increase in magnitude as the temperature anomaly increases in magnitude. And we know, of course, that thermal environments are actually becoming increasingly anomalous. So what I'm showing you here is not meant to convince you that global warming is happening. I hope we're all sort of on that page already, but these are in the transplant data themselves. And so based on the year of study, the temperature conditions were increasingly sort of deviant from historical averages in the studies that were conducted in the more recent years. So, you know, as we go on and do experiments in the field, we are doing them under increasingly anomalous conditions, and that is evident in these transplant data, not just the global record, but in the data set for local adaptation as well. It's not the case for precipitation, but it is the case for temperature. These transplant studies are being done under warmer and warmer and warmer conditions. Okay, so when we look at local adaptation then as a function of whether the experiment was done under conditions that sort of match the historical average of the local population or temperatures that were at least 2 degrees warmer than the historical average, we can see that it really does affect the outcome of the study. So when you're done under historical average conditions, then the local advantage is strong, and it's strong no matter what the foreign population's historical climate was, whether it was warmer or cooler. But when you do a transplant experiment in these more recent years under anomalously warm conditions, we see now that the local population still has a strong advantage over cool foreign populations, but they've lost it. They no longer have a local advantage over these warmer, sort of pre-adapted foreign populations. And this is now despite that separation between the local and foreign populations, all those other things that made the local populations locally adapted, they are starting to matter less and less. They are causing these foreign populations to be about just as fit. When we have temperature going in one way and the sort of biotic interactions and other things about the site are sort of being overturned by these temperature effects. And if we look at the probability then that the local population is the best in the set of whoever was transplanted at the site, that probability goes down and down and down as these temperature anomalies get stronger and stronger. And if we think about another way of examining local adaptation, which is sort of, do you do best at home or do you do best elsewhere? The probability that your home site is where you do best also gets lower and lower the stronger the temperature anomaly is at home. So this is really demonstrating that as that climatic rug gets sort of pulled out from under these populations, they are losing their home site's advantage, and populations from elsewhere are the ones that are best matched under extreme conditions. Okay, so to wrap up this section of the talk, there's sort of a glass half full and a glass half empty interpretation here. So the glass half full interpretation is when I look at these Gaussian curves for fitness as a function of temperature, I think, you know, wow, fitness is actually really robust to large temperature deviations, 5 degrees, 8 degrees. Relative fitness is still actually quite high before you get into those sort of shoulders of the curve. So fitness is actually really robust to large temperature deviations. Nonetheless, when we look at the data, it tells us that local adaptation is actually slipping away, right? As these temperatures deviate from historical baselines, the local advantage that populations used to have is decaying, and they are no longer best fit in their own sites. And that's despite all of the other things that made them fit there that aren't climate related. So that is concerning. And I think the big question is, you know, that given strong selection, how much connectivity is there to move adapted alleles in? What is the relationship between declines in relative fitness and declines in absolute fitness? Does this mean demographic collapse, or does this just mean strong selection and the opportunity to sort of evolve in real time under that strong selection? These are the big unknowns, I think, when it comes to what these results actually imply. Okay, so I want to turn to the second part of the story, which is to think about how local adaptation varies, not over time, but now across space. And this is sort of returning to some of those original themes in my, my lab's research about geographic variation in adaptation. And so the question here is not, does local adaptation decay over time, but does local adaptation change over space? So if we think about the source and the sites where populations are being moved, does being geographically central or climatically central versus marginal explain some of this variation in when and where we see local adaptation? And there's a lot of hypotheses out there in the literature about how this geographic variation might be structured. So on the one hand, people have hypothesized that populations at the edges of a species' geographic or climatic range should be less locally adapted because they lack genetic variation to respond to selection, and that's how the range sort of gets pinned by maladaptation. And so this is probably an overly complicated graphic to illustrate the point, but basically theoretical models focus on how this sort of interplay of maladaptation causes small populations to be inundated with maladapted alleles, which makes them small, which makes them prone to drift, which makes them unable to respond to selection. You sort of get these feedback dynamics that create maladaptation, where at the center of the range, populations will evolve local adaptation along a spatial or climatic gradient, whereas at the edges of the range, you might get populations that are inundated with maladaptive alleles and cannot adapt to the local conditions. So this would predict that local adaptation is lower at range edges compared to range cores. Another idea is that range boundaries are not at equilibrium with the environment, and particularly at sort of leading edges that have recently expanded. These populations are younger and they might have had less time to locally adapt to the conditions where they're residing. There's also a good bit of theory about how these sort of sequential founder events can let even deleterious mutations rise to high frequency through drift. And so these populations might harbor expansion load that would make them somewhat maladapted. And on the other hand, though, there's hypotheses, particularly at sort of older, sort of trailing or sort of historically refugial edges, that populations might actually be more locally adapted. So they're often in extreme environments where there's strong selection. They might be fragmented and able to evolve in isolation, and they might have been stuck there for a long time and therefore older and more locally adapted. So we have these sort of very contrasting hypotheses that make contrasting predictions about patterns of local adaptation across space. So for these sort of equilibrial range limits held in place by maladaptation, we would expect that edges are less adapted than centers. If we have a non-equilibrial or sort of recently expanded range, we don't really have an expectation for the trailing edge, but the leading edge should be less locally adapted due to drift and load and time. And then in these extreme or isolated environments, we would predict the opposite, that these edge populations that are actually more strongly locally adapted to their fragmented and extreme environments. We can make a set of companion predictions about population quality. So in these, all of the hypotheses actually predict that our edge or leading populations should have lower quality, so lower average fitness independent of the environment because of things like drift and inbreeding that happen in either under expansion mode models or with extreme isolation or small size. Okay, so we set out to test these predictions in our database of local adaptation studies. So again, we're using the same transplant studies with the same fitness data. We're still looking at local adaptation. So all of this is, as I've already explained in the prior sort of section of the talk. The only thing I want to introduce now is this estimate of population quality. And so I'm going to go back to the white bark pine example, again with the same design where we've got 2 gardens and these different populations planted across them. So population quality here, we're just estimating as the average performance across sites, where we take the performance of any given population, let's use the red one, and it's planted in 2 sites. And so we first normalize the red population's performance to the average performance of all genotypes in the blue site, the average performance of all genotypes in the red site. And that's to get get rid or sort of normalize across differences in site quality. We might have highly productive sites or really sort of sites where everybody does poorly. And so we're going to normalize to that baseline difference in site quality. And once we've done that, then we can just say the average quality of the red population is its average performance across all the gardens in which it was planted. And we can do likewise for the green population in the 2 sites, the blue population, et cetera, just the average performance across sites. Okay, so the other piece of this we need though is the range position. And so to do that, we had to do a completely separate data gathering step where for each species, we searched the Global Biodiversity Information Facility, which is a sort of repository of all herbarium and other sort of museum collections of occurrence records of a species. And for each species, we mapped its distribution. And this work was done by a sort of partially overlapping, but also a lot of new folks who came in for this project, including 3 folks who are currently in my lab, and then Julie Leah, who was a postdoc at UBC at the time. Okay, so for this, here we have the occurrence records for the white bark pine in the dots. You can sort of draw the polygon around them as a very sort of coarse approximation of the species range. And then we can find the center of that polygon to define the center of the range. And we can look for each source and each site at sort of where it fell within both the geographic and the climatic range of the species. And so that's shown here for the geographic position. We can estimate sort of what is the latitude of the source of this source population relative to the mins and the maxes across which the species has occurred. Or we can do it sort of in this based on the polygon where we have the distance to the center and the distance to the edge as a metric of centrality of the given population. And we can do this as well for climatic space. Again, for each point, we can extract its climatic values from these sort of climate products, and then characterize each source and each site in the distribution of temperatures that the species occurs in across its range. Was it sort of central within conditions the species often experiences, or is it getting out towards the tails of extreme temperatures relative to the rest of the species distribution? Okay, and so we can model then either local adaptation or deem quality as a function of these geographic predictor variables, our climatic predictor variables, some covariates that I'll sort of ignore, but I'm happy to answer questions about, as well as, I forgot to put it on the list, the temperature anomaly relative to historic conditions based on our other results. And I'll just, this is a pretty terrible slide. I'm happy to answer questions about the analysis. But suffice it to say, we're modeling our response variables as functions of these geographic and climatic predictors to answer these questions. Is local adaptation stronger or weaker at range edges? Is population quality lower at range edges as predicted by these hypotheses that I presented earlier. Okay, so we're going to start with the geographic position. So here's sort of relative latitude, 0 being central to the species range, positive values being towards the poleward edge, negative values being towards the equatorward edge. Local adaptation is our response variable. And then this is the one based on the convex polygon, where you're either at the core of the species range or at the periphery without regard to direction. So this could be towards the east, the west, the north. This is a sort of directionless metric of peripherality. Okay, and so when we look at the predictions or the fitted values, we can see that there's support for a decline in local adaptation from these equatorward edges towards their poleward edges. So there's not support for a unimodal relationship here, but support for basically a linear decline as you move from south to north. And there's no support for the directionless metric having any effect on local adaptation. So it's really the latitude that it's explaining the spatial variation, not one that is agnostic with respect to direction. When we look at the same response variable as a function now of climatic range position, are you sort of cool or warm or central with respect to temperature? Are you dry or wet or central with respect to precipitation? We see no effective precipitation that we can detect, but this strong sort of U-shaped pattern with temperature, where we have stronger local adaptation in thermally extreme sites, whether they're cool extreme or warm extreme. Local adaptation tends to be enhanced when you're in a thermally extreme site. And I want to point out that these thermally extreme sites are found sort of everywhere. They're not just at the range edges. So if we look at the thermal position and the latitudinal position of these points, polar, equatorial edges are not necessarily hotter. Polar edges can even be quite hot. There is a correlation here because there's bazillions of data points, but you can decouple space and temperature pretty readily across a species range. So we're finding these pockets of climatically marginal sites kind of everywhere. Okay, and finally, last result here, I want to look at population quality now. So the same setup here, we've got latitudinal range position and temperature range position, but now our response variable is that population quality, the average performance of a population across sites. And so here we see that populations that come from the range center tend to be of higher quality across sites than populations that come from either edge, whereas populations that come from hot places tend to be of low quality compared to populations that come from cooler places. Okay, so let's put this all together. If we sort of stack the results for local adaptation and deemed quality on top of one another, we can see that populations from northern edges tend to be not very well locally adapted. So they don't have a home site advantage, and they do sort of poorly everywhere, be it at home or away. And so this is really consistent with genetic load. It's consistent with having either drift and inbreeding or perhaps expansion load from recent range shifts, but it's not consistent with them having been there and sort of stably locally adapted to their environments. Now, on the other hand, when we look at southern populations and populations from warm places, whether they're at the edge or not, they tend to be highly locally adapted, but sort of poor on average. And so our interpretation of this is that they seem to be actually highly specialized, perhaps with trade-offs. So they're doing really well in their extreme sites, but they're not necessarily doing well elsewhere. And so this is consistent with sort of time and isolation favoring specialization. So if we step back, I hope what I've sort of at least started to convince you of is that by synthesizing data across all of these sites, we can start to sort of explain when and where local adaptation isn't ubiquitous. And so what are the things that are causing populations to fall out of this locally adapted quadrant and into these other conditions, whether it's temporal shifts in the environment that's sort of pulling the rug out from under them, or spatial processes, sort of eco-evolutionary feedbacks across time and space that are causing populations to be, say, universally inferior. We can start to understand this variation in the strength and the prevalence of local adaptation over time and space. And I just wanted to end by sort of, I think local adaptation is fantastic, and I sort of forget to justify why studying it is important. But I do want to sort of zoom out and think about, like, why does it matter if local adaptation is weakening or is sort of absent in a certain place or over time? And, we think of biodiversity often, it's easiest to start with species diversity, but, genetic variation within species, and particularly adaptive genetic variation, is a key and sort of more cryptic component of biodiversity. But it's one that's incredibly important for fitness across a range of environments, for buffering populations from temporal change. And it's often thought to be a sort of key precursor to things like ecological speciation that would actually lead to higher level orders of biodiversity. So it's a key component of biodiversity, and I think understanding causes are important in its own right. But these results also do have some implications for more sort of hands-on conservation applications. So one thing that people are often tasked with doing in a sort of restoration context, conservation application, is to choose source populations. And the mantra is that local is better, local is best. And finding that populations are experiencing adaptational lags, they're actually mismatched to their current environments, and there are other populations elsewhere that are equally, if not better, adapted to future conditions, has really important implications for how we would go about choosing populations for restoration, or even things like assisted gene flow or assisted migration. There's also been quite a bit of historical debate about the value of marginal populations, whether they're marginal in physical space or in environmental space. And these results suggest that it really depends on what kind of margin you're talking about. So northern marginal populations seem to be not very well locally adapted, and actually perhaps full of genetic load. And so the conservation value of a single northern edge population might be quite low, but a regional set of northern populations that could sort of be interbred and maintain local adaptation to non-climatic aspects of their environments while masking genetic load might be a sort of a better approach. And likewise, if you want uniquely climatically adapted populations, maybe you need to find them based on climate, not based on physical space, because those two things are actually quite decoupled. So hopefully what I can convince you that understanding these sort of spatial and temporal aspects of local adaptation can help us think about not just the spatial and temporal landscape of biodiversity, but also how we might go about making choices to conserve it. So I will stop there. I hope I haven't gone on too long, because I'd love to take questions if there's time. Oh, but before I do, I need to acknowledge all the work that the folks that I've listed prior have done on these analyses, as well as both Joe Herford and Chris Eckert, who shared some of their databases for other projects that we built on. And yeah, and you for listening. So thank you so much.
Cool. Thank you so much, Amy. That was super interesting, a great talk. And just to remind everyone, please either type your questions in the Q&A and I can read them out loud, or raise your hand and we can unmute you and you can ask questions for Amy. And I see already here one hand, and it's Victoria Sork. She also had questions in the Q&A. I guess that she's gonna ask them out loud.
Okay, thank you. Amy, in really wonderful talk. And I'm just really amazed that you and your group were able to compile all this data and get it to analyze it. I have a few questions. One is all about how you did it. So one is, you say performance, population quality. I'd like clarification of how you were able to measure that for all of these studies. And in terms of filtering these, the studies that you used, could you repeat again the qualifications for them? Because I'm also like, how on earth did you get deviation, yearly deviation in climate compared to warmer? So sorry, these are questions in the weeds, but so for instance, it seems to me it'd be very hard to get a tree species, versus an herbaceous, like looking at the year that something is measured for an herbaceous plant might mean a lot, but for a tree, it might not mean at all. So maybe you could just help me understand how you set up all of the filters, because the results you got are really, really exciting and important.
Well, thanks. And yeah, I glossed over a lot of the details, so I'm sorry for that, but let me, I'll try to tackle them, and maybe you can just let me know if there's other things that I'm missing. So the climate deviations, I'll start with that one. What we did was we defined the historical sort of baseline, the 51 to 1980, that was the same for everything. The experiment conditions were tailored to the study and the data. So if it was an annual plant and they did it in one growing season, then we used the months of the growing season under which they measured fitness. If it was, say, a provenance trial and they measured survival of trees over 10 years, we took the 10-year average of their experiment. And so that sort of smooths out anomalies, right? Because we're sort of taking an average over longer periods, but it reflects the duration over which the experiment conditions happened relative to the lifespan of the organism and the lifespan of the study. We also played with doing things like what was the single most extreme monthly temperature in the experiment, no matter when it occurred, thinking that that was sort of that imprint of selection might be the thing. And it didn't actually really matter, but so yeah, everything is scaled to the duration of the study and the results I showed. In terms of performance and fitness, we required the study to have a demographic estimate of fitness. So I'm sorry, we threw out stuff that only had size as a metric, which is debatable, but it did restrict the studies we looked at. So it had to have survival, recruitment, reproduction, or some combination thereof. We have that fitness type in as a random effect. So if studies that look at survival tend to find more or less local adaptation than studies that look at a composite measure, that's in there to adjust the intercepts. We tried to rank them, what's the best estimate of fitness, and use only the best from a given study, but that was a little bit fraught because after composite, which is sort of maybe clearly desirable, it's sort of harder to say is recruitment better than survival or vice versa. So we put it all in there and let it be a random factor.
So what's
I think you asked me other questions. Yeah, go ahead.
What's the difference between fitness, performance, and population quality?
Oh, fitness and performance is just me being a bit sloppy. So fitness, so everything that went in as a response variable was either survival, reproduction, recruitment, or a composite thereof. So if they had lambda or lifetime seed production over several years, cumulatively, that's in there. So that when I say performance, I'm just sort of being sloppy and interchanging that synonymously with these fitness components or fitness proxies.
Okay.
Population quality is different. So we use the fitness data then to estimate either local adaptation, which is the ratio of local to foreign fitness, or population quality, which is the average fitness of a population across all the sites to which it was planted after normalizing for site level differences in average performance. So one is a relative comparison of a local population to a foreign, and the other is just the average population's performance regardless of site, not contrasted to other things.
Okay, so I'm now going to ask a more general question, and then I'll cede the floor because probably everyone would like more general questions. In terms of seed sourcing, if plants aren't locally adapted, what advice would you, based on what you know so far, about what kind of seeds you should use for assisted migration under conditions of increased climate warming?
Yeah, that's a tricky one. I mean, so I think, it would be tempting to say, like you have a climate project, you have to define your time span. So what are you trying to sort of boost adaptation for? Is it for 20 years from now or 80 years from now, right? And then you would have to sort of, you'd have a population of different GCMs that would probably give you a range of different scenarios. And let's say that then you could then, for each given one, you could find a population that best matched that. That's already pretty fraught, right? Because you don't know what things are really going to do, and you don't know that a single population would be a best match for any of them. And that's only looking at climate, which is estimated with error. So I guess I lean towards sort of this portfolio approach of like, we're not smart enough to know, but we just need to throw a lot of variation at the mix, let selection and recombination happen, and just give them the biggest sort of bolus of variation for selection to act on. Which is kind of a cop-out, but I think anything else is assuming we know more than I think we do about everything that's important and everything that's going to happen. I don't know, I'd be really curious to hear your thoughts in response though.
Well, I'm going to let other questions and you and I can exchange, but I think your Northern Edge data actually don't support that interpretation because they show that those which do have sort of conglomerate and they do bad everywhere.
Right, but if you were going to choose a southerly population or set of populations, which one would it be? Right, so that's where I just start to worry that we don't want to get too,
Okay.
But yeah, I agree. The northern populations don't, and that's on average, right? That's just on average across hundreds of species. So then the other thing is that for any given application, you just want to drill in and do the thing that's actually supported by the data for your species if you have it, right? Because that's an average pattern across lots of disparate things.
Thanks. I'll let other people throw in their questions.
So great. Thanks, Victoria. And there's a question here by Sam, and he's asking if do invasive species ever apply to this type of work, or does that surpass the definition of a foreign population? So I guess that he's asking if in these studies people think about invasives and what happens in that case.
Yeah, I mean, so that's sort of the most foreign one could get, isn't it? We threw out studies that were done on the non-native range of a species or source populations that were brought in that might have naturalized elsewhere, and if they were brought back into the species native range. So we excluded all of that because we were trying to look at things like historical levels of local adaptation and how they were changing. You know, arguably evolution's happening quickly, and there's evidence that species have evolved even in their new ranges. So I think it might be interesting to sort of add those in at some later time, but I don't have any inferences about them right now because we did filter them out.
Cool. I have a quick question about in some of these analyses, or in all of these analyses that you showed us, you're basically looking at only at abiotic factors, right? Like temperature, precipitation, and stuff like that. What happens? I mean, I guess that it's very complicated to start adding, I don't know, pollinators or seed dispersers or, I don't know, soil bacteria and stuff like that. Has anybody looked at this and how that will potentially change some of these adaptations in the local edges or the central parts of the populations once you start sort of adding interactions or biotic interactions? Is that possible even to think about in a modeling framework that it's doable?
Well, we've tried. It's not wasn't terribly satisfying, but I'll tell you what we did. So there's basically a third analysis that I didn't present, but it was led by Anna Hargreaves, and it's out in AmNat, so I can sort of share the reference. But what we hypothesized that the studies that didn't detect as much local adaptation might have been ones that had filtered out key biotic interactions. And often what people do is it's just such a brutal amount of work to do these reciprocal transplants in these common gardens. And so like you plant all these seedlings and then you put up a fence because, oh my God, please don't eat this, you know, huge amount of work that I've just done. But if adaptation to herbivory is part of local adaptation, well, you've just taken it out, right? So what we did was we categorized studies by whether they ameliorated the biotic environment or not. Usually it was in the form of things like fencing or other things to keep out herbivory, but there were all sorts of other, you know, changing the soil type, adding Anyway, so we categorize them by whether they manipulated the biotic environment or the abiotic environment or both. And our hypothesis was that the less you did to mess with the environment, the stronger local adaptation would be. And if you took out the biotic component, you might be undoing the signature of local adaptation. And we did not find much support for that at all. And so it was actually quite surprising. Now, do I think that things aren't locally adapted to the biotic environment? No, but you know, maybe we just need to do more careful experiments. And it was usually only ameliorating negative interactions, so usually weeding and fencing. But we don't have, you know, much evidence at all about local adaptation to mutualisms from the data that we have.
Yeah,
This wasn't studied.
very cool. I guess there's no more questions, and I have maybe perhaps a last one, and I let you go. I guess that in that meta-analysis that you did, it's not possible to look at actually kind of genetic data to assess for actually movement of alleles or actually gene flow and stuff like that. But perhaps in your own work, has anybody in your lab, or have you been looking at actually quantifying truly movement of genes and quantifying genetic diversity across the ranges and stuff like that, that sort of give us a kind of a genetic perspective on local adaptation and potentially adaptive alleles in different parts of the geographic range and movement of those alleles?
Yeah, it's that's something we're just dipping our toe into. So we're a postdoc in the lab, Daniel Anstad, is like literally variant calling right now so that we can start to map geographic variation in alleles that are associated with climate. So it's brand new to us, but it is, you know, the sort of landscape genomics is a field unto itself. And you know, obviously folks like Victoria and others have been doing this for much longer than we have. And
that's
yeah,
great.
I have more to say than I do.
Cool. No, just I was wondering because of you know, this amount of experiments that you have done and all these huge ranges. That would be super cool. But anyway, so I think that there's no more questions here. And thank you again, Amy, for joining us. And I'll let you go to your next meeting. I think you have your link for the next meeting with students,
Okay.
right?
Sounds good. Yeah, thank you so much. And I will see, I guess, some of you over on the other Zoom.
Great. Okay, thanks, Amy. Bye.
Bye-bye.